Hello everyone and welcome back for more human physiology. In the past two videos we have talked about water and its capacity to form hydrogen bonds and how that influences the types of solute particles that water is of course capable of interacting with, and that's going to have a lot to do with the sorts of molecules and other things that you're going to find in your aqueous bodily fluids like intracellular fluid, blood plasma, etc. So that's a very important fundamental principle for us moving ahead in this human physiology class. Now, what we're going to talk about in this video is something that still has to do with water and still has to do with aqueous solutions, but something that's just a little bit different. We're going to be talking about the concept of pH and buffers. So let's go ahead and get into this to find out what this is all about. Okay, so water itself can actually dissociate into ions. So in the previous video, we saw that if you add salt, sodium chloride, to water, that salt will dissociate into sodium ions and chloride ions. Well, water can do this too. It doesn't do it very frequently, but water can dissociate into what are called hydrogen ions. This is just a hydrogen atom that has lost its one valence electron, so all it really is just a proton, since most hydrogen atoms don't have neutrons, so sometimes you'll see this hydrogen ion called just a proton. And then also hydroxide ions. You can see that if you add them back together, you get all the atomic components of a water molecule. Like I said, water does not do this very frequently at all. If you have a beaker of water, the vast majority of it is going to be intact water molecules. You're not going to find hydrogen ions or hydroxide ions in very high concentrations at all. In fact, if you've got a beaker of pure water with no impurities or no solutes or whatever, the concentration of each of these ions is going to be 1 × 10⁻⁷ moles per liter. So very small concentrations indeed. Well, what this is going to lead us into a discussion on are acids and bases, right? So acids and bases are just substances that, if you add them to water, they have the capacity to change the concentration of either hydrogen ions or hydroxide ions. And what we're going to see here is that both of these concentrations will be changed at the same time, just in opposite directions. So an acid is a substance that when you add it to water, it causes the hydrogen ion concentration to increase, and the hydroxide ion concentration will go in the opposite direction. It will go down. And then on the other hand, a base will do the exact opposite. A base will decrease the hydrogen ion concentration at the expense of increasing the hydroxide ion concentration. So what we're really looking at here is a balance between hydrogen ions and hydroxide ions. Acids will shift the balance in favor of hydrogen ions, and bases will shift the balance in favor of hydroxide ions. However, if you multiply the two concentrations together, it will always equal the same number, which is 1 × 10⁻14. So this is always a balance that should have a predictable outcome. So what pH is just a convenient measure of the hydrogen ion concentration in an aqueous solution. That's all it is. It's just totally there out of convenience to give us a general idea of how many hydrogen ions are present in a given solution. So the pH scale runs generally from 0 to 14. 0 being very acidic, meaning there are a lot of hydrogen ions, and 14 being very basic, indicating that there are not very many hydrogen ions, but there are a lot of hydroxide ions. So technically it is possible to have a solution that is below 0 or above 14, but the concentrations of the hydrogen and hydroxide ions get so extreme there that it's not really going to happen in biological systems. So for our purposes, we can just stick with the 0 to 14 scale here. Now, if you look at the way we calculate pH, you'll notice that it falls on a logarithmic scale, a negative logarithmic scale. So you can calculate the pH by taking the negative log base 10 of the hydrogen ion concentration. So if you're a wizard with working with logarithms and exponents, you can actually calculate the hydrogen ion concentration if all you know is the pH. Now, the good news for us is that we are not going to really be utilizing this equation. We're not really going to be doing any calculating because this is not a chemistry class. But what I do absolutely want you to appreciate is the significance of the logarithmic scale here. Basically, what it means is that if you have a difference of 1 pH unit, that indicates a 10-fold difference in the concentration of hydrogen ions between the two solutions. If there is a 2-unit difference, then that would mean a 100-fold difference. If there's a 3-unit difference, that would indicate a 1,000-fold difference. So you can hopefully kind of see where we're going with that. Let's take some of the examples here in the figure that shows us examples of the pH of different substances and where they fall on the scale, and let's use this as an example to work with. So our first example, if we look at blood, which we are going to be finding out quickly, blood likes to maintain a pH of around 7.4. If we compare the pH of blood at 7.4 to the pH of something like urine at a pH of 6.4, well, if you compare those two numbers, there is exactly a 1 pH unit difference between blood and urine. Blood has a pH that is exactly 1 pH unit above the pH of urine. That means that blood is 10 times more basic than urine is. Or if you wanted to say it the other way, you could say that urine is 10 times more acidic than blood. So urine, therefore, would have 10 times the concentration of hydrogen ions, and blood would have 10 times the concentration of hydroxide ions. Another example we can work with here, if we look at black coffee, which is fairly acidic at a pH of 5, and we compare that to the pH of pure water, which is 7, in that case, if you'll remember, in pure water, the concentration of hydrogen and hydroxide ions is equal, meaning that 7 represents something that is purely neutral on this scale. Neutral meaning that there is no clear advantage between hydrogen and hydroxide ions. But for this comparison, since coffee and water are 2 pH units apart, that indicates a 100-fold difference in the concentration of hydrogen and hydroxide ions. Coffee has 100 times the concentration of hydrogen ions. Pure water has 100 times the concentration of hydroxide ions. So the reason why we ultimately are going to want to be aware of this and to be able to keep track of this is that we don't want to forget that all of our bodily fluids are aqueous in nature. They use water as the solvent. Now, we've already seen how different fluids are different in their chemical composition. So, for example, intracellular fluid has a lot of potassium, extracellular fluid has a lot of sodium. But ultimately, because they are water-based, they are going to have associated pH values. And we're going to come to appreciate in the next few chapters that just like with anything else, like blood pressure, blood glucose, body temperature, etc., the sorts of homeostasis metrics that we talked about in Chapter 1, we also want to keep the pH of our bodily fluids well regulated as well. Our bodily processes will not work nearly as well if the pH of different fluids like the blood are all out of whack. So how can the pH of bodily fluids change as it pertains to body processes? Well, in Chapter 1, we introduced the concept of metabolic reactions, both catabolic and anabolic reactions. Well, there are thousands upon thousands of different types of chemical reactions that happen inside the cells of your body. We lump those all together and just call them your metabolism. Well, some of the molecules that are consumed and produced during your metabolic reactions, we call those metabolites. They can actually themselves act as either acids or bases. So if you do consider all of these chemical reactions happening inside of your body, at any given time, some of those chemicals that are being produced or being used up can change the pH of your bodily fluids fairly drastically. So just as with anything else, we need to make sure that we have some sort of homeostasis counterweight in place to make sure that the pH of our bodily fluids do not fluctuate too terribly much. Now, there are several different ways that we do this, but one of the front line and most important ways that we do this is the utilization of what are called buffers. A buffer is a simple mixture of different chemicals that will steady the pH by taking on excess hydrogen or hydroxide ions as they are produced. So if a particular metabolic reaction starts producing a lot of hydrogen ions, these buffer molecules can actually react with those hydrogen ions, take them on and absorb them so that those new hydrogen ions do not contribute to the pH value. So one of the most common buffering systems, not the only one, but one of the most common and effective ones in the human body is the carbonic acid and bicarbonate system. Carbonic acid is H2CO3 and bicarbonate is HCO3-. So not that it's terribly important, but this is an example of a buffering system that uses a weak acid, which is carbonic acid, and its conjugate base. The bicarbonate is what is left over after the carbonic acid gives up one of its hydrogen ions to the solution at large. So in this case, as hydrogen ions are produced, those bicarbonate ions can actually take that hydrogen ion and reconstitute themselves as carbonic acid. Or if hydroxide ions are being produced, the hydroxide ion can steal off one of those hydrogens from the carbonic acid, and instead of changing the pH, we just increase the concentration of bicarbonate. So the nitty-gritty details of how buffers work are not super important to us right now, but we will become more intimately familiar with them as the semester goes on. But ultimately, the important thing here is that for something like your blood plasma, which for reasons we will come to appreciate much later on in the semester, we want the blood plasma to have a pH of just about exactly 7.4. There's not really a whole lot of leeway on either side. So, for example, metabolic reactions like those that occur during cellular respiration will absolutely try to make the blood more acidic. So buffers really represent an important way to make sure that that does not happen. So let's take a little bit of a closer look specifically at cellular respiration on the next slide. But one last little reminder here is that you should keep in mind that buffers can only be effective within very narrow pH values, usually a range of about 2 pH units. So if the pH of a bodily fluid gets too far out of whack, then buffers aren't really going to be able to save you if the pH is outside of their effective buffering range. Okay, so let's take a little bit of a closer look at cellular respiration. Cellular respiration is something that we're going to look at in a lot more detailed fashion in Chapter 3, so I'm not going to say too much about it right now. But essentially what you see right here is kind of how we handle carbon dioxide molecules that are produced during cellular respiration. So that's one of the main things that cellular respiration does. It makes a lot of ATP molecules that our cells can harness and use for energy, but one of the byproducts of cellular respiration is carbon dioxide and then also water. Well, here's the deal. Carbon dioxide and water can actually combine together in a chemical reaction to form carbonic acid. You can ignore this kind of carbonic anhydrase signifier right here. We'll learn more about this enzyme a little bit later, so don't worry about it too much right now. All you should be thinking about right now is that every time cellular respiration happens, you dump off carbon dioxide into the blood. And as carbon dioxide starts to build up into the blood, it turns into carbonic acid, which can then dissociate into bicarbonate and hydrogen ions. Well, every time this happens, we are increasing the concentration of hydrogen ions in the blood. So, if you're keeping up here, that sounds like kind of a problem. So here's the question. As the concentration of carbonic acid increases, what's going to happen to the pH of the blood in this case? Well, if you said that the pH will decrease, you are absolutely correct. The more carbon dioxide there is in the blood, the more of that carbon dioxide is going to be converted to carbonic acid, and the more carbonic acid there is, the more hydrogen ions there are going to be, and the pH will start to drastically decrease. So in addition to buffering, another thing that we have to do in this case, especially with carbon dioxide, is we have to make use of the respiratory system to get rid of some of that excess carbon dioxide, since we've already established that these carbon dioxide molecules are indirectly decreasing the pH of the blood. So once we get to chapter 22 and we start looking at the respiratory system, you're going to come to appreciate how good and how important it is for us to have a mechanism for eliminating excess carbon dioxide from the body, not just because it's a waste product, but because it does very drastic things to the pH of the blood and other bodily fluids. Okay, that's going to do it for this video and this discussion on pH and buffering. We are certainly not done talking about the importance of pH and buffers. We'll talk about it later on when we talk about protein structure and function. We'll talk about it way down the line when we start talking about the respiratory system and the blood, but that should be a pretty good primer on the subject for now. So thank you for your attention, and I will see you next time, and we will start talking a little bit more specifically about chemical reactions in the body and the importance of molecules called enzymes, which act to speed up those chemical reactions. So thanks again, and I will see you next time.